Good evening and welcome to today's session in our immunology teaching program. Today's teaching program will deal with host-pathogen interaction from an immunological perspective. To tell us more about this, we have with us Dr. Anjana Thakur ma'am, who is a senior scientist, ICMR postdoctoral scientist and senior demonstrator at PGI Chandigarh. Dr. Anjana Thakur ma'am is
Thank
has
you so much. Thank
completed.
you.
Ma'am?
I'll take it from here. Thank you so
Yes,
much.
ma'am. I think ma'am has been with us over the last several lectures in this series, so she requires no introduction. But it's a great pleasure to have you here, ma'am. And without any further ado, I hand it over to you.
Thank you. So, hello everyone, and good evening, and welcome to the today's session on host pathogen interaction. So, in today's this lecture, I'll be touching upon bacterial and host interactions. So, let's begin. We will be discussing about definitions and dynamics of host-pathogen interactions, then a role of microbiota, pathogen entry portals, and modes of transmission, virulence factors like adhesions, invasions, toxins, evasions, etc. Then various host defenses like innate and antibody mediated, and then also we will talk about the mechanism of immune evasion by pathogens. So if we talk about the dynamics of host microbe relationship, it is very diverse. And I will be mentioning only these three terms here. Like one is mutualism, where both host and microbes benefit each other. And example for this is gut bacteria, which synthesizes vitamin, various products for the use of host. And like when I'm saying host, we are talking about human here. So they synthesize vitamin K, vitamin 12, which is used by host, and they get nutrition from the
Yeah,
host
yeah.
for their survival.
Huh. I have ordered.
And okay, so host, when we say host and microbe, so microbes are basically, or the pathogens, or these are the microorganisms basically, and the host is the one where these microorganisms are either doing the beneficial association or causing the disease in. So then mutualism we just discussed. Commensalism is like where one partner is benefited and another one is like neither helped or nor harmed. So example for this is skin commensals like Staphylococcus epidermidis, which live on us but cause us no harm. Then there is parasitism. In case of parasitism, so it is, as the term suggests, that microbes benefit at host's expense and cause harm to the this host by causing disease. Examples of the pathogens, these parasitic pathogens are like Plasmodium, Mycobacterium, tuberculosis, Chlamydium, etc. If we talk about the this normal flora or human microbiota, then this refers to the collection of microorganisms that naturally live on or and inside the human without causing disease under normal conditions. This includes almost all the microbes like bacteria, fungi, virus, protozoa, etc. And main habitats of these human microbiota are uh, our skin, then oral cavity, respiratory tract, gastrointestinal tract, and then this genito-urinary tract. So here are the picture. These pictures are just showing. In the first picture, you can see these are the different bacteria which reside at different locations. Like if we are talking about vagina, there is lactobacillus, and in skin, so as I said that epidermidis is there. There are other like bacterial species also there, and then in the respiratory, then they're in the oral, and they're in the gut. So if we see that this second picture, this is again the elaboration of the gut microbiota. So it is showing the different bacteria living as an association with the human in different parts of the gut, like jejunum, then ileum, then colon, then appendix, and duodenum, and stomach, and esophagus. So we can see here variety of microorganisms. Some are common in all of them, and some are different. This is again showing just the distribution of the microbiota. Like we can see that on skin there are epidermidis, then corynebacterium, etc. Then in oral cavity, and what they do there, they basically compete with pathogen, produce antibacterial substances, and tolerate dryness and salt. So another is oral cavity. In oral cavity, we have Streptococcus mutans, Neisseria species, Actinomyces. And here, they sorry, their major roles are like they form dental biofilms, initiate digestion, and may cause dental caries if overgrown. Then there are respiratory tract infection, upper respiratory. These are caused by Staphylococcus aureus, Streptococcus pneumoniae, and Neisseria species. Then usually these are harmless in nasopharynx, may become pathogenic in lungs or sinuses. So these can easily go to the and become the endogenous infections in favorable conditions. Then there are gastrointestinal tract where E. coli, bacteroids, and lactobacillus and this enterococcus are there. And these aid in digestion, and as I said that they synthesize vitamins also and protect uh, against the pathogens. Then there is vagina, there is lactobacillus again. It maintains the pH, and then urethra is there. There are Staphylococcus epidermidis and Enterococcus fecalis. So it may cause, if it increases there, then it may cause UTI. Now we talk about the functions of the microflora. So, or normal flora. So first is that these are protective. How these are protective to the host? So these compete with the pathogens and for the nutrients and binding sites. Hence, because they are already present there in the host, so it is not easy for the foreign microorganism to come and adhere to the surfaces. So these are giving the competition. Then there is they also produce antibacterial substances, and again they are also helpful in maintaining the environmental condition in the host, like low pH, etc. Then metabolically these help in the digestion and synthesize vitamins, influence energy absorption and metabolism. Then these immune system, they can also do immune system modulation, like they can help in the or stimulate the development of immune tissues like gut associated this lymphatic tissue and lymphoid tissue, and this mucosa associated lymphoid tissue. So it also induces production of secretory Ig antibodies and promotes immune tolerance. How it helps the immune system distinguish between self and harmless microorganisms. So because these are already present there, so our immune system is already familiar with the kind of antigen it has. So that's why it helps in the distinguishing in the self and non-self. Then there are structural or maintenance barriers. They also maintain that. How? By strengthening the epithelial cell junctions, promote mucus production, occupy epithelial surfaces and form physical barriers against the pathogens. But if there is dysbiosis means imbalance in the microflora. If there is dysbiosis, that may lead to chronic inflammations. It has been found that germ-free animals show underdeveloped immune organs, and such organisms have increased susceptibility to infections and allergic diseases. So there are various factors which can affect the normal flora. First is age. It actually changes with the age. Then there is diet and nutrition, depends upon the our diet and nutrition. So every individual would have different this microflora or normal flora. Then there is antibiotic use, then can also cause dysbiosis and overgrowth of can and this biases can help in the overgrowth of opportunistic pathogens like this Clostridium difficile and Candida albicans. Opportunistic pathogens are those which are otherwise commensals living there in the body, but when they are getting favorable environment, they become pathogenic. Other factors are like hormonal changes. So if there is increase in the estrogen, that favors the lactobacillus growth. And if lactobacillus growth is there, so there is acidic pH in the vagina. But during the menopause, there is a hormonal imbalance, and that may reduce the protection by this, which is given by the acidic pH. Then there is hygiene and environmental factor. Like you cannot excessively sterilize things, or poor hygiene can also cause the imbalance. Sorry. Then overuse of antiseptics or antibacterial soaps, that can also reduce the beneficial this flora. Then there is also immune status and health conditions, like in case of immunosuppressions like HIV, chemotherapies, when somebody is taking chemotherapy, or if somebody is diabetic, in that case also it can these commensals can overgrow and act as a opportunistic pathogens. Then there are infections, and then they can also, sorry for a moment. Okay, so various infections and inflammations can also alter the microbial populations, and that can again give chance to those opportunistic pathogens to grow. Now, bacterial, if we talk about infection, basically, when we are talking about bacterial infection, so if we define this infection term, this is basically the invasion, evasion, and multiplication of the microorganism within the host tissue and cause the damage to the tissue. So these bacterial infections are, or these bacteria which cause infection are of basically two types, that is extracellular and intracellular. Extracellular are those which are coming from outside, like Salmonella, Shigella, Yersinia. Sorry, these are facultative, like Vibrio cholera, Pseudomonas, and E. coli. These are extracellular. Then we have intracellular. Intracellular bacteria can also be divided into two categories. One is obligate and another one is facultative. So in case of obligate and intracellular, these are completely dependent on the host cell, and they are basically living inside the host cell where they are growing and multiplying. They cannot grow and multiply outside of the host cell. Examples are Rickettsia, Chlamydia, Coaxilla. And facultative intracellular, these bacteria are Salmonella, Shigella, and Yersinia. they can grow inside also, and then they can grow outside of the body also, host also. So this process of infection has various stages, and these stages include transmission from an external source into the portal of entry. Then from the portal of entry, it actually invade, and and there is evasion of primary host defenses like skin or stomach acids, and finally it enters the body. And adherence, after that, it adhere to the mucous membranes, and then colonization by growth of bacteria at the site of adherence. After that, there is toxin production, and that invasion is accompanied by inflammation. Then there is host responses, including both innate and adaptive, during the this first three, step third, four, and five, where the adherence, colonization, and toxin production is occurring. Then finally, progression or multiplication of the organism, and progression of the disease, and finally, resolution of the disease. So, before going further, let's just have a look at the various host defense mechanisms against bacteria. So, we have innate and adaptive immunity. So, in innate immunity, we have various barriers like physical barriers, chemical barriers, etc. Like physical barriers, we have skin, mucous membrane, cilia. Then chemical barriers, we have lysosome, we have stomach acid, antimicrobial peptides, etc. Then we have cells also, innate immunity cells, which are non-specific in nature, like neutrophils, macrophages, dendritic cells. Then there are there is complement system also, which cause opsonization, because of which actually facilitates phagocytosis. Then there is lysis of bacteria, in especially in case of gram negative, and then there is recruitment of inflammatory cells. So this is all what does innate immunity does, cells and the organs does. And then we have adaptive immunity, which is basically antibody mediated, or like both cell mediated and antibody mediated uh, this immunity. So what happens in adaptive immunity? We can see that if antibodies neutralize toxins, and we can see here more clearly in this picture. Once the antigen is coming inside the cell, so first is that toxins are acting there. The toxins are being neutralized by the antibodies. We can see here. Then complement. After that, there is complement mediated lysis. Like these are the complement proteins. Like C3b is there, which is actually interacting with the antigen. And then there is opsonization and interacting with this complement. This protein is interacting with it, and taking it to the complement pathway, where finally it will form the membrane attack complex, and form various pores in the membrane of the bacteria, and finally kill the bacteria. Then there is opsonization and phagocytosis. These almost have a similar fate. And then there is anaphylatoxin mediated mast cell degradation. So mast cells are granulated cells. So here again, when there is a like antigen, so we can see here these are the components, or we can say proteins of the complement system. They actually helps again in the like they take up the antigen to these cells, and finally these cells release the various enzymes and mediators, which actually cause the inflammation, or cause the damage of that particular organism. Then we have macrophages here. So when there is tissue damage, we know that there are various there is chemotaxis also there, because of the various chemical substances which are releasing around the injury and injured tissue. So because of that, this adjacent that blood vessel the endothelial cells of this gets vasodilated, and through their endothelial cells, their junction of the endothelial cells, they actually loosen up here, and from here these cells like neutrophils and macrophages release from this blood vessel, and they finally reach the site of the that injury, and they again engulf the this is the macrophage, they engulf the antigen, and this is how they process this is the fate of the antigen once it enters the body. Then there are various virulence factors like adhesion factors. Adhesion factors allow pathogen to attach to the host cells. These are like pili, adhesions, proteins, viral spikes. Then there are invasion factors, like they enable entry into our movement through tissues like invasions, enzymes like hyaluronidase. Then there are toxins like those disrupt host cell function or integrity, the exotoxin like cholera toxin, endotoxins like gram-negative LPS. So we know that there are exotoxins and endotoxins. Exotoxins are those which are released by which basically both gram negative and gram positive, specifically exotoxins are characteristic of gram positive, but they can be secreted by gram negative also. But endotoxin, it's specifically it's specific to the gram negative only, and it cannot be uh, expressed by gram positive bacteria. So then there is immune evasion factors like capsule is there, antigenic variations are there, various proteases are there, and then there are like biofilm formation is there, and various secretion systems are also there. So these are the various portals of entry. So when pathogen, there is after transmission, pathogen reach to the portals of the entry, a pathogen entry. So we have various these portals. These are similar as that of the organs of the or innate immune system. So these are like components of the innate immune system, like respiratory tract, gastrointestinal, skin, genital tract. So all of these are portal of pathogen entry. So this table is just showing the list of the pathogen which can enter through various routes and cause various diseases. Like in respiratory tract, it is Streptococcus pneumoniae, and it causes pneumonia. Then Haemophilus influenza is there, cause meningitis, and there are viruses also like rhinovirus, then Epstein-Barr virus, infectious. These call common cold, then infectious mononucleosis, and similarly, in case of gastrointestinal tract, there are Shigella dysentery, which cause dysentery, then Salmonella typhi, which cause typhoid fever, and others are there. In case of skin, there is Clostridium tetani is there, rabies virus is there, etc. Then in case of like Staphylococcus aureus, for Staphylococcus aureus also skin is the entry portal, and then there are genital tract like in this Neisseria gonorrhoeae, Treponema pallidum, and Chlamydia trachomatis. These are again these also this genital tract is the entry portal for these pathogens. So so in transmission, what happens that there are various types of transmissions, like person to person is there, which can be by touch or by sexual contact or by droplets like from cough and sneeze, or it can be airborne, small droplet nuclei, etc. Then it can be indirect also, like for mites, like coming from the contaminated surfaces, then vehicle, food, water, food, blood transfusions. Then there are vectors like mosquito and ticks. Then there are zoonosis, animal reservoirs like rabies via bites, and then Lyme diseases via ticks. So, and then in addition to that, there are heterogenic like transmission resources also. So for that, these come from medical procedures like surgery or catheters. There are two types of transmission. Horizontal is there and vertical is there. Horizontal is what we have just described, that these organisms are coming to the portal of entry and they are entering there. And transmission modes are various, like self, human to human can be there, animal to human can be there, etc. But this is just a list of various organisms which are coming from various means, like from the direct contact. Gonorrhea comes from the direct contact. Then uh, dysentery coming from the is not coming from the direct contact. It is from the fecal-oral route. And then there are there is bloodborne, like hepatitis B, and then there is soil source, tetanus, and again, animal sources are there, like Lyme disease is there, hemolytic uremic syndrome is there, caused by E. coli, one of the E. coli strains. But there is vertical transmission also. So vertical transmission is the transmission when it is coming from the mother to the child. So it is transplacental, and it organisms which takes part in it. These are Treponema pallidum, Listeria, monocytogenes, and then cytomegalovirus, parvovirus, and Toxoplasma gondii also. So diseases, there are various diseases these cause in the neonates, and then there is within the birth canal also, or at the time of birth also. So these are Streptococcus agalacti and Streptococcus other species like group B Streptococcus, E. coli, Chlamydia, Trachomatis, and others. Then there is bacteria that can come from the breast milk also, including Staphylococcus aureus, cytomegalovirus, human T cell leukemia virus. So these can come from here. Now let's talk about the adhesions. So once these organisms or these extracellular organisms reach to the portal of entry, they try to adhere there. So molecules mediate adherence to cell surfaces are called adhesions. These are pili, capsules, or glycocalyx. But it has been found that mutants who lack these capsule, pili, and glycocalyx, they lack these mechanisms. So these are often non-pathogenic. If we take the example of Neisseria gonorrhoeae and E. coli, their pili helps in the attachment of these microorganisms to the urinary tract epithelium. And then there are glycocalyx of Staphylococcus epidermidis and certain viridin streptococci. They adhere to the endothelium of heart valves. Now, biofilms. Biofilms are basically this is a protective matrix basically formed by the bacteria itself, and it is extracellular polymeric substance. This matrix is basically extracellular polymeric substance which is made up of polysaccharides, then proteins, and this DNA. So these are coming from bacteria itself, and bacteria is forming this. Bacteria has evolved this system of its protection from various encounters. Like we'll discuss, I'll enlist them later. So first, let's have a look that where these biofilms actually occur mostly. So these occur on the foreign bodies like prosthetic joints, prosthetic heart valves, and intravenous catheters. But in addition to that, they can also form on native structures like heart valves. Then there are these biofilms are resistant. Now, what are the benefits actually they give to those bacteria? They actually resist antibiotics and host immune defenses. Like they resist antibodies or neutrophils, etc. Moreover, they also retard the wound healing that leads to the chronic wound infection and example is the in case of diabetes. Then they also cause the persistence of Pseudomonas in lungs of cystic fibrosis patients, and in the formation of, they also helps in the formation of dental plaque. So now let's talk about the invasion. After addition, there is invasion. So how this is happening? There are enzymes. Bacteria produces these enzymes like collagenase, hyaluronidase. These degrade collagen and hyaluronic acid and allow the bacteria in subcutaneous tissue. So they are specifically important in cellulitis caused by Streptococcus pyogenes. And then there is coagulase, which is uh, which is the characteristic of Staphylococcus aureus. So it what it does actually, it helps in the clot formation, and it forms fibrin from the fibrinogen. And it is also produced by Yersinia pestis, and that Yersinia pestis cause bubonic plague. Then immunoglobulin proteases. So bacterial like invasion, during the invasion, bacteria also produces various immunoglobulins, immunoglobulin proteases that can break those immunoglobulins, like and Neisseria gonorrhoeae and Haemophilus influenzae and Streptococcus pneumoniae produce IgA proteases. So these IgA proteases break down the IgA polypeptide, and this inactivate IgA at the mucosal surfaces. So that helps these organisms to attach to the mucosal surface of the host. Then there is S. pyogenes, Streptococcus pyogenes, which produces an enzyme that cleaves IgG heavy chains and reduce opsonization. Actually, because IgG causes the opsonization, when it cleaves it, so it reduces the opsonization, and so there is no activation of complement system, and hence enhance the virulence of this organism. Then there are various evasion factors, like capsule is there, cell wall proteins are there, leukocytins are there. So capsules are like these are formed by bacteria like S. pneumoniae, Neisseria meningitidis, and H. influenzae. This prevents the phagocyte from adhering to the bacteria, and also these like if there are anti-capsular antibodies, those allow more effective phagocytosis to occur. Then their vaccines contain the capsular polysaccharide of the organism as the antigen to induce anti-capsular antibodies. So basically what is happening, that the antibodies are producing in these cases are basically anti-capsular. So these anti-capsular antibodies are being used in the vaccines. So then let's talk about the cell wall proteins. Then there are cell wall proteins like M protein from the group A Streptococci asparaginase, which is anti-phagocytic. Then there are protein A of Staphylococcus aureus that binds to the Fc portion of the IgG and prevents the activation of complement system. Then there are leukocidins. Leukocidins are known to be formed by MRSA, and Penton Valentine leukocidin is the one example. These are pore-forming toxins, and these degrade the membrane of the neutrophils and macrophages. So this is just showing the various mechanisms where bacteria can prohibit the host defenses. So we can see here biofilm formation is there, biofilm matrix is there, then T cell proliferations, then there is this superoxide dismutase is there, catalase is there, then antiphagocytic this staphylococcal protein A is because of the staphylococcus protein A, like that blocks the phagocytosis, and then there are other proteins which are inhibiting the adhesions. So this table is showing the surface virulence factors important for bacterial pathogenesis. So these are in gram-positive cocci, streptococcus, these are polysaccharide capsule is there, then streptococcus pyogenes, M protein is there, and they have multiple serotypes. In case of streptococcus pneumoniae, they have 84 serotypes. So, and the thing is that when the strain of the serotype of the streptococcus pneumoniae cause a disease, the antibody which are developing which are developing in the host are only for that particular serotype. So when the another serotype is attacking the host, so there is no antibody for that, and in that way, this streptococcus pneumoniae can cause multiple infections. So there are others like gram-positive rods are there, like Bacillus anthracis. Then there are gram-negative rods like Haemophilus influenzae, Klebsiella pneumoniae is there. They also have a polysaccharide capsule. Then E. coli has protein pili, etc. So there is in N. gonorrhoeae, so its pili has which is composed of or consists of protein pillin. So this pillin has a very highly variable structure because it have a region where the amino acids are like highly variable, and so this structure is very variable, and that amino acid variation is because of the multiple gene arrangements of its coding sequences. So, because there is enormous antigenic variation that increases the pili binding capacity to the epithelial cells. And the continual changes in the pillin sequence also allow the organism to evade neutralization by IgA. So these are various like host immune responses to bacterial infection and bacterial evasion mechanisms. We have just spoken about this Neisseria gonorrhoeae. Then there is like this phagocytosis by this proliferation is the another infection process where antibody and C3b mediated. C3b is again complement that component or protein where antibody and this C3b mediate opsonization, and evasion mechanism is production of surface structures like polysaccharide capsules, M proteins, and fibrin coats that inhibit the phagocytic cells. Mechanism for surviving within the phagocytic cells, and induction of apoptosis in macrophages. For example, Shigella flexneri. Then there is complement-mediated lysis and localized inflammatory responses. These are generalized resistance of gram-positive bacteria to complement-mediated lysis, and then there is insertion of membrane attack complex. It is membrane attack complex is the one which that complement system lead to finally. So this membrane attack complex basically forms the pores in the bacterial membrane and cause the bursting of the cell. So insertion of this membrane attack complex prevented by long side chain in the cell wall of LPS. So those gram-negative bacteria, which has they have LPS, lipopolysaccharide. So that lipopolysaccharides has a side chain, is a long side chain. So that chain actually inhibit the insertion of membrane attack complex. Then invasion of host tissues. It is again AB-mediated agglutination. So mechanism is secretion of elastase. So this that inactivates C3A and C5A proteins of the complement system. Now this elastase is secreted by Pseudomonas. Then there is toxin-induced damage to host cells. it is neutralization of toxin by antibodies. This is secretion of hyaluronidase, which enhances the bacterial invasiveness. Now, there are mechanisms which are used by the intracellular bacteria to survive and grow inside the cell. So we were talking about the extracellular bacteria so far. So if we talk about the intracellular bacteria is that, as I think, yeah, I have already defined that these are the bacteria which grow inside the and multiply inside the host cell only. So their mechanism used by these to survive and grow inside the cells are like one is inhibit fusion of the phagosome with lysosome. So these are because when they enter the body, they are engulfed by the phagosome, engulfed by the macrophages where they form the phagosomes. So these phagosomes are do not fuse with the lysosome. And why? Because these intracellular cellular organisms has such mechanism that they prohibit that uh, fusion of the lysosome with the phagosome. So these organisms and by this these organisms avoid the degradative enzymes in the lysosome. So because there is no fusion, there won't be no leak of enzymes to the phagosome, and there won't be any degradation. Then second is inhibition of acidification of the phagosomes. So another thing is that it inhibits its phagosomes acidification and reduces the activity of the lysosomal degradative enzymes. Then there is third one is that escape from the phagosome into the cytoplasm, and that was done by Listeria species. So these mycobacteria and Legionella, these follows the first mechanism, that is inhibit fusion of the phagosome and lysosome, while as I said, that Listeria species follow this phagosome escape from the phagosome into the cytoplasm mechanism. So this is the Listeria, as we have just mentioned in the previous slide. This is the Listeria monocytogenes. So it actually escapes from the phagolysosome to the cytoplasm for favorable environment for their growth. Because in the phagosome, if it stays there, so there will be enzymes which are going to uh, degrade this microorganism. So these are the macrophages we can see here, and they form the phagolysosome by engulfing the Listeria, this monocytogenes. And after this, a listeriolysin is produced, which is a pore-forming toxin that forms the pores into this macrophage, and finally, these gets these bacteria gets into the cytoplasm of the cell, where it gets a favorable conditions and also becomes resistant to the various oxidative attacks. Then there's evasion by intracellular organisms, like another organism, that is Mycobacterium avium. So in Mycobacterium avium, it blocks the it again blocks the lysosomal fusion with the phagolysosome, and some mycobacteria are resistant to the oxidative attack that takes place within the phagolysosome. So we can see here, so what actually happens in this case, that once bacteria like macrophages engulf it, so there can be oxidative attacks and some of the cells can die, but some of the cells are resistant to the phagolysosomes. So after those cells survive and inhibit the fusion of this lysosome and phagosomes, and finally causing the forming the resistant phagosome. So we can see here, these are the reactive oxygen species. Various reactive oxygen species are there, but this this organism has become resistant to the this oxidative attack by detoxifying the reactive oxygen and nitrogen species. So it uses catalase and peroxidases for breaking down those this reactive oxygen species with the help of enzyme superoxide dismutase. Then there are now immune responses as can act as a contributor to the bacterial pathogenesis. So so far we just saw that how the bacteria was acting against the immune system, but now once the bacteria is there, immune response is there. Now that immune response itself can cause the bacterial pathogenesis or can cause the disease. So pathogen stimulated, what happens in this case, that pathogen stimulates overproduction of cytokines, and that leads to bacterial septic shock, food poisoning, toxic shock syndrome. And this happens in the case when there is accumulation of large number of these intracellular microorganisms in the cell. So cell wall, what is happening in this case, like cell wall endotoxins of some gram-negative bacteria activate macrophages, and these macrophages release high levels of interleukin-1 and TNF-alpha. So these interleukin-1 and TNF-alpha are in lesser amounts are good for the cell, but when there are in high levels, they can cause the septic shock. And in case of staphylococcus or staphylococcal food poisoning and toxic shock syndrome, so there are exotoxins that function as superantigens. And superantigens actually activates all the T cells that express T cell receptors and cause the overproduction of cytokines by activated Th cells and causes many of the symptoms of these diseases like this. So we can see here, this is the superantigen and on this APC. So we can see there how it triggers this exaggerated T cell activation. This is non-specific T cell proliferation, and this causes, because there is exaggerated response, it causes immune system dysfunction and finally causes toxic shock syndrome. So now there is the ability of some bacteria to survive intracellularly within infected cell can result in chronic antigenic activation of cell-mediated immunity, that is CD4 plus T cells, positive T cells, or we can say T helper cells, leading to tissue destruction by a cell-mediated response with the characteristic of a delayed type hypersensitivity reaction. Moreover, cytokines secreted by these activated CD4 positive cells or T helper cells, these can lead to the extensive accumulation and activation of macrophages that result in the formation of granuloma. Now, bacterial infections are mostly of two types. One is this pyogenic and then this granulomatous. So most of the intracellular infections cause granuloma, and most of the extracellular these organisms are like pyogenic. So, but granuloma is specifically characteristic of these intracellular pathogens, and because it helps in their survival inside the cell. So, the localized concentration of lysosomal enzymes in these granulomas can cause extensive tissue necrosis, and that can lead to the tissue damage. Like tissue necrosis is there, of course, tissue damage. So, similarly, this Mycobacterium tuberculosis can also cause the tissue damage due to the cell-mediated immune response. So, and let's talk about the diphtheria, corynebacteria. We can see here in the picture that the active subunit of this is catalyzing the basically catalyzing the addition of this ADPR. ADPR is adenosine diphosphate ribose to the target protein in the human cell. So ADP, we call this process ADP ribosylation. So now what happens in the human cells? Okay, let's talk about it. So now what this does actually, this modify the target proteins and inactivate. It can either inactivate it or either hyperactivate it. Now, what actually it does? So we'll take the example of diphtheria toxin and pseudomonas toxin. So this ADP ribosylation actually, this this actually inhibit the or replace the this factor, block the elongation factor 2. Elongation factor 2 is basically the translocation factor in the this protein formation process and in the translation process. So it is very useful because in the translocation, what happens that when there is on the tRNA, when there is one polypeptide is forming and the another like amino acid coming in, then that polypeptide is shifting to the another that tRNA. So for that translocation, this EF2 is very important. So now this ADP ribosylation actually blocks that translocation, and if that translocation is not there, then protein synthesis will not be there. So this is how this diphtheria toxin inhibit the protein synthesis in the host cell. So again, cholera toxin and E. coli toxin, these activates signaling molecules like GS protein by this ADP ribosylate and ribosylation, and then this increase the activity of adenylate cyclase activity, and consequently it actually increase the amount of cyclic adenosine monophosphate. So that leads to the production of watery diarrhea. So here we can see that Vibrio cholerae is there. So it is here interacting with the GS signaling molecules and proliferating the formation of cyclic AMP, and then finally leading to the formation of dysentery. Then there are pertussis toxins. These are ADP ribosylates, GI proteins. This is inhibitory proteins, and these inactivates like this pertussis toxin ADP ribosylation of this GI protein actually inactivates the protein synthesis of this signaling pathway. And then inactivation of the inhibitory G protein turns on adenylate cyclase again, and it causes an increase in the amount of cyclic AMP, and it actually causes the symptoms like whooping cough. So this is in case of this whooping cough. So we can see here pertussis toxin is there, and ADP ribosylation of the G inhibitory, this cascade, and then finally causing the increase in the cyclic AMP, which leads to the whooping cough. So here are various important bacterial exotoxins like Corynebacterium diphtheria cause disease diphtheria, and its mode of action is inactivates the elongation factor 2 by ADP ribosylation. Then there are like botulism is there. Then there is, which is from the Clostridium botulinum. Then there are pseudomembranous colitis from the Clostridium difficile. And there are other like anthrax, Bacillus anthracis, etc. Okay. Then there are gram-negative rods also like E. coli. They again cause the watery diarrhea, and they again toxin of this E. coli actually stimulates the adenylate cyclase by ADP ribosylation, and stable toxin stimulates this guanylate cyclase. Finally, there is no toxoid vaccines for most of them, only for the Clostridium botulism and Corynebacterium diphtherium and Clostridium tetani are present. So that will be all for today's session on host pathogen interaction between bacteria and host. So these were the references from the review of medical microbiology and immunology by Warren Levinson and immunology by QB. Few of the resources were from the internet. Thank you so much.
Thank you, ma'am. We do have one question.
Yeah, sure.
Uh,
Sure.
if considering that gut dysbiosis plays an important role in both infections and disease causation, would regular probiotics help in preventing infections or the like?
Actually, probiotics are actually known to increase the or maintain the balance there, because if we see that our diets, dietary and nutrient that intakes are not that adequate actually. So somehow we almost um, all of us face that dysbiosis at some levels, at different levels, we can say. So to balance that, I think those probiotics are good to take, and studies says that they are very helpful. So yeah, that's a good choice also.
Thank you, ma'am. We have no further questions. I thank you for taking time out of your schedule to give this session. I'd also like to thank everyone who joined us for today's session, and would like to remind you to kindly take up the quiz on the same topic available at MediEdu TV. Thank you, everyone, and thank you, ma'am.
Thank you so much. Thank you.
Thank you, ma'am.
Okay, thank you. No more questions. Shall I quit at
Yes,
this?
ma'am. Yes.
Okay.